All right, so we actually have two products. So it makes a little bit more maybe confusing of what our company is, but I'll try to do my best to explain it. So firstly, we're a company which has purchased one of the devices, and the other one we have also purchased, and we're developing it further and further. So we were established in the 2019 in the Czech Republic, and part of our company is in Brno, in the Czech Republic, and the other part is in Paris. So we are located in two different countries, and we do have two different systems. We're a very small company, I think, for at least for what I have seen. We're up to 30 people, not up to 30 minutes. We specialize in optical instruments, which focus basically on lifestyle imaging. So what we want to do, or the mission of our company, is to provide the best possible solutions for lifestyle imaging, for two technologies. One is quantitative phase imaging, and the other one is super resolution. Our one machine maybe was developed by Tescan company, which is an electron microscope company. Perhaps some of you are familiar with it. I have already mentioned, yes. So our focus, as I have said, is the lifestyle imaging. What we do is that we do everything in-house. So from software development to assembling the optics, and everything is basically done in two different locations. And what we try to do is not only just to develop products, but we try to take hard part in joining the research. So we collaborate a lot with other universities when we work together on different grants on how to develop and push our technology further, because we believe that it is still sort of underrated, at least for the quantitative phase imaging, because the adoption in comparison to other technologies, as we see it in the industry, is rather slow, but it is very robust. And so what we offer is not only products, but always also help with analysis, or we go do biological experiments, and try to now push the AI move also into the advanced imaging analysis. But I'll just a bit more about it later. So first, let me introduce our first machine, which is developed in Brno. So this is machine is called Q-Phase. It is a quantitative phase microscope. Basically, it uses one source of light, where the light has a different phase, and the phase goes first through a reference sample, and then also through this pointer. So first we go split the light, and we go through a reference sample, and then we go also through the sample. So we do have two sets of similar objectives, and the incoherent light source which is passing through is then joined together and constructs a hologram, which you're able to turn into a QPI image. This is the QPI image which comes out, and this QPI image is unique in a sense that it carries a lot of information. So it's not only an image by itself, but each pixel has a weight, and therefore you can calculate many things. So what we provide, as it says in the name, is quantitative data about your sample. So this is one of the main advantages of our system, is that we provide quantitative data such as the thickness, and then you can easily calculate, for example, dry mass of a cell. You can do this real time with our microscope, or when you're doing the analysis, then you can observe this data, and you need to later on wait for the calculation. We do have different sets of objectives. It goes from the magnification of 4 to 60, and recently we have developed also a sort of motorized stage that helps you I think we can do it at 6 by 6. I think we can do 36 well plates right now. So that is some of the addition that we have. So this is the sort of how our microscope looks. This is the more of a in-depth look inside with the laser path going through, just to give you the idea. You don't need to have the same thickness of the sample. Maybe I should have mentioned that. But you need to put for the reference sample, you need to put glass there for it to be at least sort of a similar thickness. The tricky part for us always is that the objectives need to be exactly the same. So for us to get the precise imaging, also see it a bit later. So what this provides the clear image is that it's very precise background discrimination, which really provides you with very good segmentation. So you can segment the cells based on their size, based on their movement. There is a very high cellular contrast for that. And you can do this automatically in our software. We're implementing an AI feature to help you segment the cells. So when you're working with large sets of cells and large cell populations, then this comes really handy. You can also do this manually. So perhaps some of you have experienced this or are working with different systems. So I think this is really one of the nice features is that the segmentation is done for you beforehand, but then you can really easily change it by yourself. So what do you get from our Q-Phase machine? So as I've mentioned before, you get the mass, the area, the density. You get different parameters. You can measure each single cell, their speed and everything, and you get the precise quantitative data for it. So also when you replicate your experiments, you can sort of add on the validity. Other things recently we have added was the speed and the motility. On the images right here, you can see again images from our software. This would be the motility diagram. You can see the cells or their movement measured. You see the numbers, so each cell has always a number in our software. The sort of in our software is that you first observe your sample, so you can then already get some real-time data. Then you use the segmentation, and then we provide also a very robust analytical software with our solution. So we probably like to work with different software for analysis. So we also have a plugin for imaging, so you can easily export the data from our Q-Phase machine and then import it into the imaging, for example. So different rates of applications. Cancer research is one of the popular ones for what is being studied right now on our microscope. A Czech researcher, Mr. Brabec, is working on the microstatics, which are basically it's a different type of cancer treatment where you wouldn't necessarily kill the cancer by inhibiting it by applying, let's say, a different type of chemicals to destroy the melanoma, but what you're trying to do is just to stop the cancer from spreading. So that's why the term of microstatics, because you sort of stop the migration. And this is a very popular research right now, which is working on different types. Basically anything which is biological or life cell imaging related is very interesting to us. Another one would be Jan Balwan, who is studying cell death. So also very popular. It's just some module specification, but perhaps more interesting, you can download the brochure on our website. So when do you need a Q-Phase? So is a complete solution, including a software from acquisition to publication. So makes it really handy. Of course, it's a new software that you need to learn to work with, but the scientists who spend the time and learn to work with our software are usually very positive about its functionality. And so we do believe it's really one of our strong points. For a small company, so the system can be upgraded anytime, and we do provide really top level service for our customers, because we really try to work hard with them. So we're implementing different changes to the software according to their needs, because we can still touch into the software quite easily and do custom solutions basically for them. We have lots of detailed statistics at single cell level, but we can also study cell populations. And basically, anytime you need a label-free or non-destructive way of observing, so that's when you would also choose a Q-phase. And as I mentioned, when it's the quantitative data which you are after. And then there are a few examples. So just from what you can see from what comes out from our machine. So you have the light mode where you control the microscope and the acquisition, and then, as I said, is the analytical part where you have the history of each cell. You can do the population gating, the segmentation, as I mentioned. So here is just a short video which is showing the apoptosis and necrosis at the same time, which quite interesting. So here you can see that the cell on the right shows the morphological features of apoptosis, while the cell on the left dies of necrosis. You see the characteristics swelling, and then the sudden rupture will occur soon. You can see the differences here in the mass, where the cell basically is going to disperse. And you can also see that there's a certain colors, because fluorescence also can be used at the same time with our microscope. So it's this multimodality. So you can set different things, so you can basically get the fluorescent microscope at the same time. You also get the quantitative phase imaging, which comes in handy because the LED light sources not really affect the cells that much, but once you're doing the fluorescent microscopy, of course, the excitation of the light does affect your experiments. You can time it correctly, and then you can change the thing. So this is source that I want to show is a monitoring and scattering media. So it's basically also one of the few QPI technologies where this is possible, because we use the incoherent LED light source in comparison to coherent light sources like laser, where you would not be able to get these images. It's a unique to Q-phase because it cannot be basically done with other light sources. Another application of virology, as I have mentioned, is you can see the multimodality, where you see the green on the right, where the cells are infected with the vesicular zoonosis virus, and each light that you see, it's what is paired from the fluorescent view. We have up to 3 filter set cubes, which can be accommodated with the Q-phase, and for each channel you can set the intervals differently. So just to conclude, the main advantages of the Q-phase machine are you can observe live cells. So it does not affect them. It's designed for long-term observations. It's not no phototoxicity, but rather very small phototoxicity, close to 0. You get a quantitative data. It's label-free, but of course you can use labels if you want to use the fluorescent module, so it's possible. It's a precise segmentation. It gives high contrast, and imaging and scattering media, and it's a bundle together with our analytical software. Now I'd like to give word to my colleague Caroline, and she will present the live coding part. She is from France, so she will take care of the French machine.
Okay, good afternoon, everyone. I'm about to share my screen, if I manage to do it. I don't think I'm allowed to. I can't.
Let me check. I'm not quite sure because I also don't seem to be able to make you a presenter first.
Okay, I'll just share my screen.
I don't know what is wrong with the Zoom. I'm very sorry.
Don't worry.
No problem. I will just share the screen and then.
So I think Caroline, I've now made you co-host, so you should be able to share yourself as well.
Let me see. Oh yeah, I have more options. Here it is. Okay. So let's do it. You should be able to see my screen now.
Yeah,
great.
Now it seems to be working.
Okay, so sorry for the little bug. So again, good afternoon. So I'm Caroline, application specialist at Telight, and I'm very happy. I don't see anything. You have a dark screen as well?
Yes.
Yes.
Oh, great. To show you, to give you a little introduction to our second project, which is called LiveCodim, and it's an add-on system to perform super resolution microscopy from any fluorescent microscopes. Maybe Michael, it could be better if you continue sharing and we
I will share and then you just wave at me.
Because here it seems to be very not working at all.
Stop the sharing.
I'm very sorry. So yes, great, thank you. So as you can see now on the image, LiveCodim, it's the box that you can see on the right of the microscope. It's an add-on system that is plugged to the C-mount of any fluorescent microscope to turn it to a super resolution platform. And by using a very unique technology based on conical diffraction, we can produce some patterns of light that are going to be focused on the samples, and then we use an algorithm to reconstruct a super resolution image from this acquisition. Next slide, please. Thank you. Can you play the video? So conical diffraction, it's an optical phenomenon. Never mind. So conical diffraction is an optical phenomenon using the properties of a biaxial crystal, which means that when you have polarized light that passes through this crystal, it emerges with some patterns of light. And by controlling the polarization of light before and after the crystal, you can decide which pattern you want to project on your sample. And here we are using laser scanning geometry, on which we introduce our beam shaping system. So from the laser source, you will then shape the beam, project some patterns of light, which are these half moons that you can see on the bottom left corner. So two lobes of light separated by a tiny region of darkness where there is no light, and these patterns will be rotated 2 times, so we will acquire 3 images for each region of the sample. And by using this galvanometric mirror, or as you would have done for a confocal, you are going to scan your sample, acquire 3 micro images per region, and then feed your processing algorithm. So LiveCodim can be defined as a confocal sim, because we create localized structured illumination really focused on the sample. And this allows us to get rid of many artifacts that can emerge with wide field SIM techniques by only focusing on tiny parts of the sample. And instead of using a PMT as you would do for confocal, we use a camera to record our images. So we have much more details that and a variety of intensity on each micro images. As we are using this laser scanning geometry, we have the same penetration as confocal, so 0.5 millimeter, which is a huge advantage compared to other super resolution systems. And we don't use any specific properties of the samples except the fluorescence. So you don't need to change anything to your sample preparation. We use a regular fluorophores, and this technique is completely achromatic, so it can be used with any commercially available dyes or fluorescent protein already implemented in your lab. Can you switch to the next slide, please? Here is a better view of the inside of the box of LiveCodim, where you have a laser source that is going to pass through our beam shaping system composed of the biaxial crystal and surrounded by a series of 2 double Pockels cells to control the polarization. Then it goes to the sample, scan thanks to the galvanometric mirror, and goes back to the camera, the big blue square you see at the center. We collect all the images and then process them through our software and algorithm to reconstruct the super resolution images. Next slide, please. Thank you. And thanks to all this, we can then break the diffraction limit and reach a resolution of 90 nanometers. As you can see on these images, on the right side of the screen, you have an example of DNA origami that has been acquired with a 488 nanometers laser source, where we managed to discriminate the 2 parts of the origamis, meaning that we can resolve 90 nanometer lateral resolution. And it's valid. This is valid on this artificial samples, these DNA origamis, but as well on real sample, real cells, as you can see on the left, where we reach almost the same resolution in this case, but this is just an example. We managed to measure the width of some membranes. Here, the outer mitochondrial membrane, about 100 nanometers. Next slide, please. And thanks to our system, again, it's achromatic and can be used to image 4 colors at the same time. So as you can see here, we simultaneously acquired several channels to resolve fine details in the cells. On the left image, we access to the organization of microtubule in orange, and we again looked at mitochondria, the outer membrane. And on the right, you have an example of some component of the nuclear pore complex depicted in blue, in light blue, and the inner membrane of mitochondria this time. This is why it looks a bit more blurry, for which our resolution doesn't allow us to access to the finest detail, but still we can reject lots of out of focus light like a confocal would have done. Next slide, please. So I didn't highlighted this part of the system, but in our box, you can either, with our system, you can either perform wide field imaging from your microscope, as you maybe are used to do, or confocal imaging, or what I prefer calling pseudo-confocal, because we use a virtual pinhole, sorry, and again, we use the properties of the camera instead of using it as a photon multiplier. So as you can see in the center, where we collected the images, project them, reconstruct the So collect the micro images during the acquisition, reconstruct the image in the of the sample, and without performing any processing. And when we process, we can really increase the resolution and the level of details that is accessible with our system, as you can see on this example. And the next slide, please. And again, our system is really dedicated to live imaging because thanks to the beam shaping technologies that we are using, we really project a very low level of light intensity on the sample, as you can see on the curve on the left corner, compared to a standard the standard light dose given by a confocal. The live CODIM is located in the green part of the curve, so very low light intensity, preventing from photobleaching and phototoxicity to cells. And you can perform long-term imaging, as we show here, by looking at the dynamics of growing mitochondria in MDCK cells. Next slide, please. And to wrap up everything, I just want to remind you that so live CODIM, it's using a scanning geometry, but with structured illumination, so that you can extract spatial frequency information and reconstruct a super result image with a resolution up to 90 nanometers. Again, it's tailorable to any fluorescent, any visible wavelengths, from 400 nanometers to close to near infrared, and it's commercially available and suitable for any commercially available fluorophores. As we use a very low level of intensity illumination, it's particularly adapted for live imaging because it reduces drastically the phototoxicity for your sample and the photobleaching of your fluorophore. And with this, I would like to thank you for your attention.